This is the third video for the unit on photosynthesis. In this unit, we will learn about the second stage of photosynthesis called the Calvin cycle. It is this stage that inorganic carbon dioxide is reduced to organic carbohydrates. The Calvin cycle consists of 15 chemical reactions that occur in the stroma of the chloroplast, which synthesize carbohydrates from carbon dioxide. The reactions are grouped into three main steps. The first step is carboxylation, where carbon fixation occurs. The second is reduction, where NADPH is used as an electron donor and a 3-carbon carbohydrate is released as a product. And the third is regeneration, where ribulose 1,5-bisphosphate is remade. During carboxylation, a 5-carbon molecule called ribulose 1,5-bisphosphate, also abbreviated RUBP, is joined to a molecule of carbon dioxide, producing a 6-carbon molecule. However, this 6-carbon molecule is very unstable and spontaneously breaks into two 3-carbon molecules called 3-phosphoglycerate, also abbreviated 3-PGA. This first step is called carbon fixation because it takes a molecule of inorganic carbon in carbon dioxide and incorporates it into an organic carbon molecule. The enzyme that catalyzes this reaction is ribulose bisphosphate carboxylase oxygenase, which is abbreviated as Rubisco. Rubisco is a slow enzyme and is produced in very large amounts in the cell. Up to 50% of the protein in the leaf is Rubisco, because of its highly important role in carbon fixation. In order to complete a full cycle of the Calvin cycle, 3 molecules of RUBP are joined to 3 molecules of carbon dioxide, producing a total of 6 molecules of 3-PGA. In the second steps of the Calvin cycle, the 3-PGA must be reduced to increase its potential energy. This requires both ATP and NADPH, which were produced during the light reactions. 3-PGA is reduced to another 3-carbon molecule called glyceraldehyde 3-phosphate, abbreviated G3P. Because there were 6 molecules of 3-PGA, we will form 6 molecules of glyceraldehyde 3-phosphate. This requires 6 molecules of ATP and 6 molecules of NADPH. G3P is also called a triose phosphate, or 3-carbon carbohydrate. 1 molecule of G3P will leave the chloroplast to form glucose in reactions that occur outside the chloroplast. The remaining 5 molecules of G3P are required to stay in the stroma to complete the regeneration stage. During regeneration, the 5-carbon RUBP needed for carbon fixation is regenerated. This is a complicated process of 12 different chemical reactions. During those reactions, the carbons of G3P are shuffled and rearranged to regenerate RUBP. This process requires 3 ATP produced from the light reactions. The best way to understand regeneration is to follow the total carbon around the cycle. 3 5-carbon RUBP started the process, providing 15 total carbon atoms. They were joined to 3 carbon dioxide, giving a total of 18 carbon atoms in 3-PGA. When 1 molecule of G3P leaves the chloroplast to form glucose, that subtracts 3 carbon atoms. The remaining 5 3-carbon G3P contain 15 carbon atoms, providing the correct number of carbons necessary to regenerate the 3 RUBP. The triose phosphate molecules exit the chloroplast and are used to make glucose through anabolic reactions in the cytoplasm. It takes 2 G3P molecules to make 1 molecule of glucose. Glucose molecules are then covalently attached together to form starch and stored in the cell as starch granules. Starch granules are formed during the day while photosynthesis is occurring. The glucose from these starch granules is used by the cell to produce energy through cellular respiration in the mitochondria. There is one problem with the Calvin cycle reactions, which involves the Rubisco enzyme. Unfortunately, the active site of the enzyme is able to bind both carbon dioxide and oxygen and catalyze the reaction with RUBP. However, if Rubisco adds oxygen instead of carbon dioxide to the 5-carbon RUBP, no additional carbon is added, producing one 3-carbon PGA and one 2-carbon molecule called 2-phosphoglycolate. When this occurs, no sugar is produced, but the ATP and NADPH are still used in order to regenerate RUBP. This results in a waste of energy for the chloroplast with nothing to show for it. This process is called photorespiration.